All right, histology, don't stress this stuff. I'm going to show you all the pictures, and by the time we're done, you'll know what they could ask. All right, it's not as bad as you think. Right, so we're looking at the nephron here. This entire thing is the nephron. We start with the glomerulus, right? There's millions of these in the kidneys. And I think it's, oh, I might get this wrong. One fifth of the blood that comes through, don't quote me on that, actually goes through the kidneys each time it goes around. But anyway, that's a lot of blood to deal with, you know, at a time. So you take these glomeruli, these millions of them, and they have little holes in it. And we'll look at those in a second. And through these holes, the urine is passed through, and things such as red blood cells are too large to go through. So we're looking at ions and proteins and stuff like that. And then through this process, through the loop of Henle and whatnot, you can pull back what you need, get rid of what you need. It's very fine-tuned process to be able to urinate the things you want to urinate and keep the things you want to keep. So we'll get into it. So you can see what they call this glomerulus is a tuft of cells, a tuft. But basically what it means is, or a tuft of capillaries, excuse me. Basically what it means is these capillaries come through, and the urine is pressed out into this urinary space, or Bowman's capsule. You can see as the blood comes in, it goes through and it's kind of pushed out. It's like filtered through. And then from there, you can get to the proximal convoluted tubule. So you should take a snapshot in your head of what this picture looks like, right? Glomerular capillaries. Of course, this space is going to be where the urine comes through. And you can see a little bit closer, these podocytes are like little foot processes that kind of make the fenestrations so that the fluid can go through here. And these slits are very small, but you could see how the urine could kind of be pushed through there, keeping the larger things like cells in your system and get rid of the smaller things. Very important to know that these are the things that are going to dictate whether the blood could get through, or excuse me, the urine can get through. Charge is a big thing. Negatively charged molecules tend to be repelled. Size also is a big thing. This is why red blood cells are kept and don't go, you shouldn't have any blood in your urine. And then shape matters too. Excuse me. So if they ask you any sort of things like this, they'll probably ask size. That's pretty straightforward. But there are also some other things that dictate what we're looking at. All right, now, in regards to the tubules, this image, this H&E stain, they'll probably ask you either proximal convoluted tubule or distal convoluted tubule, okay? So you need to be able to tell the difference. The way I tell the difference is that the proximal convoluted tubule has about 5 nuclei, okay? It's kind of sparse, okay? Versus what we look at when we see the distal convoluted tubule, there's a lot of nuclei and it's tighter, okay? So the proximal convoluted tubule, one of the major goals of the kidney is we don't want to lose protein, right? You should never have protein in your urine either. You want to keep your protein. So it's going to pull back all the protein. Glucose too. You don't want to lose glucose if you don't have to. So you're going to pull back all the glucose. Now, diabetic patients have so much glucose that it overwhelms the system. So if you get glucosuria, right, glucose in your urine, maybe you're thinking diabetes. Sometimes it happens in kidney failure too. So these are the things, pretty much urine should be sterile. It should be composed of urea, mostly water, stuff like that. But if you have proteins or glucose in there, you need to investigate a little bit further. All right, so proximal convoluted tubule, again, you can see these nuclei are kind of sparse, right? You can see around here. Right, and also if you're trying to differentiate looking at proximal to distal, you'll see a lot more proximal convoluted tubules than distal convoluted tubules, okay? Now, we'll get to distal in a second and I'll show you them next to each other so you can differentiate these. But the next thing is this different view, this longitudinal view of the loops. Now, there are 3 things that I think that they could ask you. The first one is this empty space. So this empty space or this lighter area that we're looking at is going to be your loop of Henle. Now, how this works is the vasa recta is the blood-containing vasculature around the the loop of Henle. Okay, so what happens is not blood, but the urine components, whether it be salt or water, can be transferred from this clear space, this loop of Henle, to the vasa recta, to the blood. Okay, so you could kind of see here these little blood vessels going around, but they just lay next to each other. Okay, so it could transfer from the urinary system to the blood, back and forth as they go around the concentration. Now, I'm telling you this for a reason, because they could ask you this clear space, which is the loop of Henle, but remember the vasa recta is blood components, it's vasculature. You see, these are red blood cells. So anytime you see them running next to each other, you see this loop of Henle here, and you see the vasa recta here. Giveaway, if you see red blood cells, it has to be vasculature, so it has to be the vasa recta. Okay, and you could see that here. Right here, you can see the loop of Henle, and you could see right here is the vasa recta, and we'll get to this one in a second. All right, again, vasa recta, right? A little bit different of a view, but you can kind of see the red blood cells that come through here. But the whole point is that they wrap around each other just so they could transfer things back and forth. So same thing here, we're looking at the vasa recta with blood, and the loop of Henle is clear. Look for red blood cells for the vasa recta. Now, distal convoluted tubule, I have some closer up views, but if you look here, you can see there's a lot more nuclei. They're right next to each other. So that's how you're going to differentiate the proximal versus the distal loop of convoluted tubules. You can see there's a lot more there, and they're kind of a little bit they're a little bit more eosinophilic, but I think I have a better image coming up. But yeah, they're a little bit smaller. Remember, they're less abundant than the proximal convoluted tubules, and they have more nuclei. So yeah, again, I just want to point out that the ratios are different. You're going to see a lot more proximal to distal convoluted tubules. Let's see if we can look at some. So I would say like this proximal, but look at this one next to it. I would say that that's distal, right? So you could appreciate the difference. Proximal is kind of looks a little bit bigger, less nuclei. Distal is going to be smaller, more eosinophilic, so a pinker stain, and more nuclei. Okay, now the macula densa is going to help to monitor how much sodium you have in your system. So remember, sodium and water go together. So if you have a lot of sodium, that could be indicative that there's a lot of water around too. So the macula densa is going to be here. And when we talk about that renin-angiotensin-aldosterone system, that's going to make more sense. But it actually lies next to the glomerulus, all right? So it kind of sits up against it, and that's just a good point to be able to monitor your sodium input. And you could see that here. So when we do physiology, we'll talk about that. I'm just trying to show you the pictures, right? So find your glomerulus, right, our tuft of capillaries, and that macula densa is going to just sit right next to it. All right, so now we could break this down. This is very important. This is a huge point for medication, okay? So your volume, the amount of volume you have in your body dictates a lot of things. It dictates your kidney function, dictates how hard your heart has to work. It dictates the vasculature if you have some sort of edema. So you just want to tightly regulate it. And this renin-aldosterone renin-angiotensin-aldosterone system helps to regulate it. Okay. So what happens is if the body senses that you have too low, okay, hypotensive or decreased blood pressure, it's going to go through two processes. So think about it. If you have low volume, so you're hypovolemic, your pressure is going to be low too, right? Low volume, low pressure. So you want to do two things. You want to save water, and you want to vasoconstrict the vessels. You want to save water to bring up the volume. You want to vasoconstrict the vessels to bring up your blood pressure, okay? So that's what we're going to do. So renin's going to be activated. It's going to go through this process of activating angiotensin. So it's going to go from angiotensinogen to angiotensin 1. For whatever reason, ACE is made in the lungs, just one of those weird things. But ACE is the enzyme that's going to convert angiotensin 1 to angiotensin 2. This is a very important enzyme because they use ACE inhibitors as a first-line treatment for hypertension. All right, so if you inhibit this, you're going to inhibit the process of making angiotensin. Okay, think tense as it squeezes, right? So angiotensin is going to squeeze down on the vessels, bring up your blood pressure. All right, so I hope that makes sense in regards to the blood pressure. Now, think about it. If you give an ACE inhibitor and you can't activate angiotensin 2, you're going to help to decrease the blood pressure, right? Okay, so that's one process. So now we've constricted the vessels, but we also want to conserve volume or conserve fluid. So we're also going to activate aldosterone. Very important. Also used in pharmaceuticals to regulate blood volume and blood pressure. So what aldosterone is going to do is at the terminal end, at the distal convoluted tubule and the collecting duct, it's actually going to use these channels, which we'll get to in a little bit, but it's going to bring back sodium. And remember, anytime we bring sodium back, we're going to bring fluid back or bring water back. So through this process, patient comes in hypotensive, or whatever. The patient is hypotensive, you're hypotensive, whoever's hypotensive. Renin's going to be activated. Angiotensin 2 is going to be activated to squeeze down on the vessels. Aldosterone is going to be activated to bring back sodium, therefore bring back water. Perfect. We brought our blood pressure up. We have more fluid. That's a good day. All right. We'll go through that again just because it's very important. But let's finish off the histology stuff. So the collecting tubule is going to be at the end after you get to the distal convoluted tubule. Okay. And you can see some of, yeah, this is a better picture. So look at this one. So now we're looking at all three in this longitudinal cut. Remember, we said this is that loop of Henle. It's clear. We find our red blood cells. We have our vasa recta. Now they refer to this as like a dirty lumen. But you can kind of see how there's stuff in it, right? So that's going to be our collecting duct. So you can look at all three of them next to each other. The clear one is the loop of Henle early on in the nephron. The vasa recta is going to have red blood cells, runs next to the loop of Henle so that they could have that countercurrent exchange. And then once you get to the end, you have the collecting duct, which is going to have like this dirty lumen. You can see there's kind of stuff in it, okay? And you can compare them here. These yellow lines, you would say that's more of a dirty lumen. So that would be our collecting duct. This clear area next to it would be our loop of Henle. And anywhere you see red blood cells will be your vasa recta. So that's the 5 things I think they could ask you. Differentiate between a proximal or distal convoluted tubule, and looking at the longitudinal cut, are we looking at loop of Henle, vasa recta, or collecting duct, okay? So you could do those 5, you should be good. This is just a zoomed in view. Dirty here, so that's collecting duct. Clean here, loop of Henle, red blood cells, vasa recta. Piece of cake. All right, now, when we get into the urinary system, that uteric bud that formed embryologically, you get into that drainage point, then we are talking about specifically this transitional epithelium. The only time you find transitional epithelium is in this urinary system. Okay, so if they're ever talking about that, you have to be talking about the ureters to the bladder to the urethra, that area. And you can kind of see it here. They have, well, this is, I think I have a better picture of it coming up. Yeah, but this is this transitional epithelium. It's kind of hard to differentiate this from like the pseudostratified and whatnot. Ideally in the stem, they would tell you you're in the bladder area and then you automatically know it's transitional, okay? And then the ureter, it has this, they call this a stellate shape, but it'll have this transitional epithelium as well. You can see the muscularis, the 3 components to it, ending with the muscularis and the adventitia. And then you get to the bladder, and this is where I wanted to point out, they have these, what's called these umbrella cells, okay? They kind of bud out into the lumen. And the reason this is like this with this transitional epithelium, the reason this is like this is because when the bladder's full, the lumen expands, right? So these umbrella cells get pushed out. So a lot of times they like to talk about these because it's very specific to the bladder, okay? Then we get to the urethra. Still, we have transitional epithelium all the way to you get to just the tip over here. And then you have stratified squamous, and it's kind of towards the outside. So just keep that in mind.